When a zygote is newly formed, the cell will continue to divide in order to make new cells. These cells will build up a fully functioning human through the processes of cell replication, transcription, translation, and epigenetics. In the nucleus, during mitosis, in the interphase stage, helicase unwinds the two strands of DNA into the replication fork. It does this by attaching at the origin and breaking the hydrogen bonds on the nucleotide bases, A and T and G and C. This separates the DNA's double helix structure into the replication fork structure. DNA has a 5' end with a phosphate group on the end and a 3' end with a hydroxyl group on the end. DNA is anti-parallel, meaning it has complementary strands. One strand will have a 5' end facing the same way as the 3' end on the other strand. When DNA is split into the replication fork, one strand will be aligned to go from 3' to 5', while the other strand will be aligned to go from 5' to 3'. DNA gyrase is a topoisomerase, which keeps DNA from winding back together, which is also called supercoiling. Single-stranded binding proteins will bind to the DNA to stop it from recoiling, which stabilizes the DNA. Primase is an RNA primer that binds to the two strands of DNA. It allows DNA polymerase to attach to strands in order to start synthesizing the two DNA molecules. Primase helps initiate DNA polymerase to add nucleotides onto DNA strands by providing a 3' group guiding polymerase in order for nucleotides to be added. The original strand of DNA, that is 3' to 5', is called the leading strand. DNA polymerase 3 can only add nucleotides to DNA from the 5' to 3' direction, meaning it will move from the 3' end on the leading strand to the 5' end. The leading strand only needs one primer because the addition of nucleotides is continuous. The original strand of DNA that is oriented 5' to 3' is the lagging strand. DNA polymerase 3 builds from the 5' end to the 3' end in direction, although that is going away from the direction of unwinding, which causes the addition of nucleotides to not be continuous, meaning the strand requires more primers. Since multiple primers are needed and the addition of nucleotides is not continuous, Okazaki fragments are created. Okazaki fragments are gaps between primers on the strand. Primase is replaced by DNA with DNA polymerase 1. Ligase joins the Okazaki fragments together by creating a phosphodiester bond between the fragments of DNA, which creates one complete strand with no gaps. DNA polymerase 2 proofreads the two strands of DNA. It finds errors, removes them, and it will replace the errors in the strands. As cells divide, DNA replication needs to happen in order for the human's DNA to be passed on to the new cells. Transcription has 3 steps to it: initiation, elongation, and termination. Transcription is creating an RNA strand from a DNA template. It is a recipe for a protein, and it carries the code of genes in DNA. The process of transcription happens in the nucleus of the cell. Transcription factors, which are protein complexes, bind to DNA where the promoter is located on the DNA. RNA polymerase binds to the promoter with the help of the transcription factors. The promoter is a nucleotide sequence which is needed to initiate transcription for the synthesis of RNA. A common promoter is called the Tata box, which consists of thymine and adenine. DNA will then unwind, and one strand of it will be used for the template for RNA synthesis. ATP will then be needed to begin the transcription process. RNA polymerase goes down the gene sequence, creating an mRNA strand. It then reads the template and uses complementary base pairs due to RNA being synthesized and not DNA. Instead of thymine being paired with adenine, uracil is used to bind with adenine. RNA reaches the end of the sequence and termination happens. The new RNA strand separates from the DNA. RNA has to be processed before it can be used in translation. On the 5' end of the mRNA, a methylated cap is added onto the end by the use of a phosphate linkage. The methylated cap protects the mRNA from being broken down. On the 3' end, a poly-A tail is added, which is a string of adenine added by a poly-A polymerase. This also protects the mRNA from being broken down. Splicing is the last step to process mRNA. Introns are cut out of the RNA, and RNA attaches the exons back together. Introns do not code for a protein, while exons do code for a protein. Introns are cut out of the RNA segments as they are non-protein coding sections. Transcription needs to happen in order for translation to happen. This is an important process for a developing baby, for a developing fetus, because it is the first step to make a protein. Translation happens in 3 steps: initiation, elongation, and termination. mRNA moves into the cytoplasm for translation. A ribosome attaches to mRNA strand at the start codon AUG. A small subunit of a ribosome attaches on the 5' end of the mRNA strand. the subunit will move down the mRNA strand until it meets a start codon, which is most often AUG. A tRNA molecule will then bind to the start codon with an anticodon. The tRNA carries an amino acid in order for protein synthesis. Different anticodons on tRNA carry different amino acids. The large subunit of the ribosome will bind to the smaller subunit, which makes a completed ribosome. The process of translation goes from the A site to the P site to the E site. The A site is aminoacyl, the P site is peptidyl, and the E site is the exit. The starting tRNA molecule will be located in the P site, which allows the A site to be matched with an anticodon on the other tRNA molecule, which will add another amino acid onto the polypeptide chain. The ribosome will continue to move down one codon at a time, which will build the polypeptide chain until it reaches a stop codon in the A site. Once the stop codon is reached, release factors will bind to the codon, releasing the polypeptide chain by adding water instead of an amino acid. This creates a hydrolysis reaction between the tRNA and the polypeptide chain bond in the A and P site. The polypeptide chain is now let go into the cytoplasm. The process of translation needs to happen for a fully functioning human to develop because proteins need to be made to carry out the human body's functions as it continues to grow. Epigenetics is the turning on and off of genes in a gene expression, which is what genes are expressed in cells. Epigenetics changes what proteins are produced in cells. Each gene codes for a different protein, meaning whatever gene is turned on and off expresses which protein will be encoded. Different environmental factors change epigenetics and genome expression in good and bad ways, such as pollutants, smoking, alcohol, working out, and your diet. Methylation is a common way epigenetic expression can be changed. It is when a methyl group is added to DNA. If there is a methyl group on the section of DNA, genes are turned off, meaning the protein they produce is not produced. If there is no methylated group, which is called demethylation, genes are turned on. This means that the protein that the gene encodes is produced. Transcription factors are environmental or cell signals that activate proteins. The proteins will then bind to the regulatory areas in the genes, which is the promoter. This will change how much of the protein will be made based on if it increases, which is turning it on, or decreases, which is turning off the gene expression. Transcription factors can also bind to RNA polymerase, which increases or decreases gene expression and transcription. Differentiation. A zygote is a stem cell. Stem cells are able to differentiate to any kind of cell. This is important for the development of a baby because it allows the fetus's cells to differentiate, forming its main structures and different cell types. When cells differentiate to become different types of cells, the cells have different genetic expressions instructing the cell what protein will be made. The genes must be expressed differently in order to differentiate the cell, meaning when genes are turned on and off in different cells, they will encode for different proteins to be made. Around 5 weeks after being fertilized, the fetus's systems and structures will start to develop. The cells in the embryo will start to divide and differentiate to have specific functions. Cells such as blood cells, kidney cells, and nerve cells will all start to develop during this phase. After 8 weeks of fertilization, embryo will develop specialized cells and tissues and begin to create most organs. Epigenetics allows humans to have different traits and features depending on if their genes are turned on and off. This will make one person different from another person. These 4 processes continue as a human grows. This happens until the day you die. What genes are turned on and off may change, but these 4 processes all stay the same throughout life.